Good afternoon, everyone. Welcome to the Bio ASGCT and FDA Scientific Seminar Series. Today's presentation will be on genome editing technologies. These are powerful tools for rewriting the code of life. Our speaker will give an overview of existing genome editing technologies and their applications, and will share a specific, more detailed example with the nucleus-free genome editing approach. I'm Virginie Eggeman. I'm co-chair of the Bioregenerative Medicine Committee. I'm also Chief Regulatory Officer at Tessera Therapeutics, a biotech company focusing on novel genome editing technologies. I will be the moderator for the Q&A portion of the seminar today after the presentation. And first, I would like to thank all of you for attending to this seminar, including all members of ASGCT, all members of Bio, and we'd like to give a special thank you to all the participants from the FDA. And second, it is my great pleasure to introduce Dr. Carter Isham, Chief Scientific Officer at Bio, who will provide introductory remarks and introduce our presenter for today's seminar. In her role at Bio, Carter manages and directs biopolicy, advocacy, research, and educational initiatives for several teams, including the emerging companies team and the science and regulatory team. Overall, Carter's mission is to ensure establishment of effective and efficient paths forward for the development and review of innovative medicines around the world. And I think we can all relate to this important mission. Carter has more than 20 years of health policy experience and a PhD in microbiology from the University of Georgia. And I had the pleasure to witness firsthand Carter's masterful policy skills during the latest round of PDUFA negotiations. And trust me, she's very impressive. So Carter, the microphone is yours.
Thank you, AV. I feel like I'm blushing. I really appreciate that. Thank you. I too just want to say good afternoon and welcome everyone to the second installment of the Cell and Gene Therapy Science Series. In this series, it's a wonderful joint effort between Bio, the American Society of Gene and Cell Therapy, and the FDA, because it brings together the scientific and regulatory community to discuss important issues to advance the development of safe and effective cell and gene therapies. So in that vein, I'd really like to thank the American Society of Gene and Cell Therapy, Bio's member companies, and the FDA, not only for their partnership, but for their continued support of this valuable series. I'm delighted to introduce our presenter, Dr. Jean Liao. Dr. Liao is the Director of Vector Development and Operations at Alexion Pharmaceuticals, and has more than 15 years of experience in the gene editing field. She has extensive experience with cassettes optimization and capsid engineering to improve AAV manufacturability and enhancing the potency of AAV vectors. It is my pleasure to turn the microphone over to our presenter, Dr. Liao.
Thank you very much for the introduction. So thank you, and great thanks to the organizers for putting this seminar together. Today, it's my great honor and pleasure to walk you through many of the key gene editing technologies in the field. They are powerful tools rewriting the code of life. What is the code of life? Since 1865, when Mendel discovered the law of genetics, scientists have put a lot of effort deciphering the code of DNA and inventing a lot of technologies, including cloning and sequencing, in order to isolate specific genes and understand their corresponding functions. In 1990, there was a very ambitious project started. It's called Human Genome Project, in order to finish the sequencing for the human genome. It lasted 13 years. So in 2003, the finished version of human genome sequence was completed. This was a great achievement of human beings. And in between, there are many different species genome have been sequenced, including yeast, bacterial, C. elegans, plant, Drosophila, mouse, and rat. All these tremendous efforts build up a solid foundation for the advancement of gene therapy, gene editing technologies. So, trying to minimize myself. Okay. So, let me see. 1 second. My slide is blocked, so okay. I would like to introduce the nucleus-based gene editing first, because they have a relatively higher gene editing efficiency. So during the cell replication, and there are double-strand break created, there are two dominant repair pathways for the DNA repair, including NHEJ, non-homologous end join. When the double-strand DNA break happened, Cu7080 protein can recognize the damaged site and started recruiting the factors for the NHEJ, including the kinase, exonuclease, polymerase, to create the compatible endpoint and for the ligase to ligate them together. And this process, their random deletion or insertion happened. And as a result, there will be indels created. Other than that, another repair pathway is HDR, the homology-directed repair. mRNA will be created into the damaged site, and then this will initiate the long-range resection and create the 3' single-strand DNA. The binding of the RAD51 protein can help the single-strand DNA search for the homology sequence and evade into the donor DNA sequence in order to trigger this D-loop formation and amplify for the repair. So as a result, you can see that the donor sequence highlighted as brown color here is integrated into the target site precisely. So these two repair mechanism has its own application. For NHEJ, because it will create indels like random insertion and mutations, and potentially it will trigger premature stop codon formation and knock out the target gene. While for HDR, because it's precise insertion based on the donor sequence introduced, so it can be applied for DNA insertion like knock-in. But if we just based on the cell itself, double-strand break event, the editing efficiency will be low. So scientists were thinking about how to enhance the event of double-strand break in order to enhance the gene editing efficiency. In 1996, zinc finger nuclease was developed. So the zinc finger motifs was discovered in the xenopus oocyte. The structure of the zinc finger motifs include two beta sheet here, and then one alpha helix stabilized by a zinc ion shown here. Each zinc finger can recognize 3 base pair sequence color-coded here. And a very clever design that scientists thought about fuse zinc finger with the FOC1 cleavage domain. It's a nucleus cleavage domain, so that the zinc finger can guide nucleus to the target site. And then the dimerization of the FOC1 cleavage domain will trigger the double-strand break in the target site. One of the advantages of this zinc finger nuclease technology is its high specificity, because because of this dimerization of FOC1 cleavage domain is required for DNA cleavage. Only when the left and the right zinc finger meet with each other, and then they will trigger double-strand break. Another advantage for zinc finger nuclease is that it's only around 1 kb. So they will be compatible for different package methods. Well, every technology has its own limitation. Because the recognition of the sequence is rely on the protein recognize the DNA sequence. And then the sequence recognition of each zinc finger was significantly influenced by its neighboring zinc fingers. Then a lot of work has to be put in order to identify specific zinc finger sequences to target to specific gene sequence. In order to conquer this complicated protein engineer work, in 2010, talons were developed. Tau is also called transcription activator-like effect motif. It was discovered in a bacteria. They have the repeating unit of 33 to 35 amino acids shown here. And each tau, they can recognize single nucleotide through these just 2 amino acids within this 35 amino acid sequence in the position of 12 and 13. These 2 amino acids can recognize specific base pair. And same principle, when the tails were fused with the fork-1 cleavage domain, it will guide the fork-1 to the target site to trigger a double-strand break. The advantage for talon is that the recognition of the DNA by individual tau is independent of the neighboring modules. So it will be like building up Lego pieces. So you don't need to have a different mixture of talons. As long as you identify specific talon, recognize the base pair, and then you can assemble them together. It also has high specificity because of the requirement of dimerization for the fork-1 cleavage domain. But the challenge for talon is that, as you can see, they have this repetitive nature of the tau's consensus sequence, which makes it more difficult to be assembled and packaged. All these technical challenges create a more effort for the scientist to invent more simpler system. In 2012, there was CRISPR-Cas9 system published. And the CRISPR system, cluster regularly interspaced short palindromic repeat Cas system, it was first discovered in bacteria and archaea, where the bacteria has its own immune defense system. And the component for the CRISPR include guide RNA and Cas protein. I use Cas9 protein as an example here. When the CRISPR complex was formed, guide RNA will drag Cas protein into the target site. And the guide RNA sequence is only around 20 base pair. Only one guide RNA sequence recognizes target sequence right upstream of the PAN sequence, highlighted in a green here. PAN sequence is protospacer adjacent motif. It can promote RNA and DNA binding. When this exact location was recognized, and then it will trigger the Cas protein and create double-strand break. There are specific PAN sequence for different Cas protein. For Cas9, the PAN sequence is NGG. So it's not hard to imagine that for CRISPR, the design is relatively simple. You just need to design these guide RNA sequence for new target. And also you are able to address target to many different loci at the same time with different guide RNAs. But also, because it's easy to create a double-strand break, so it's easier to create HEJ, meaning the random insertion or deletion, compared to HDR, a homology-directed recombination, which is the precise editing. And the guide RNA potentially it might guide the Cas9 to a different location other than your target site. So there's a relatively higher risk of off-target effect. Another challenge for CRISPR-Cas9 is that the size is relatively big. The size of Cas9 itself is already around 4.5 kb, which has relatively limited choice for what is the package system that they can use for delivery. So I still remember during my postdoc, when we need to generate talent, we have to collaborate with specialized lab to create talents for us. But for CRISPR-Cas9, we can just simply adapt that in our lab very efficiently and handle the cloning and started to do the editing directly. Just because this system is so easy to be adapted, right after 2012, there are a boost of the development of CRISPR-Cas9 variants technology right afterwards. So not like the traditional Cas9 drive double-strand break, as I mentioned, if there's double-strand break, the NHEJ repair pathway will be preferred other than HDR pathway. But nickase was created. So this nickase can be guided by the guide RNA to the target site and create single-strand break. And if there's single-strand break, then this will trigger a more efficient way to use HDR pathway for the precise editing. In addition to that, dCas9, the dead Cas9 was also created by introducing mutations in the catalytic domain in the Cas9. The dead Cas9, it means that it can be guided to the target site, but without creating any double-strand break. So the dCas9, if they are guided into the target site, because of the structure of the Cas9, it might function as an inhibition of the target gene. Another option is to fuse dCas9 with a repressor in order to suppress the gene expression in the target site. Similarly, dCas9 can be fused with the activation domain in order to enhance the gene expression. In addition to the modulation in the DNA, scientists also fuse dCas9 with the epigenetic modulator. And using the epigenetic modulator, for example, the DNA method transferase to modify the methylation of the DNA, or histone modification to change the epigenetic status of the gene in order to control the gene expression. But all these systems are inherited from CRISPR-Cas9 system itself. So they have their inherited pros and cons similarly. A few years afterwards, base editing was developed. And the base editing is, in addition to the guide RNA and with nickase, it also fused with deaminase enzyme. I use adenosine deaminase enzyme, for example. So these nickase can be guided to the target site by the guide RNA, and adenosine deaminase can recognize the A in the target sequence. And then it will deaminate these A to inosine. During the DNA replication, polymerase will recognize these I as G. So as a result, we can observe that adenosine deaminase fused with these nickase and guide RNA, they are able to change A to G in the target site. That's a single base pair change mediated by this base editor. Similarly to adenosine deaminase, using cytidine deaminase, it can change C to T. The biggest advantage of the base editing is that it doesn't generate double-strand break. So if we imagine that if we enhance the efficiency of double-strand break, they might trigger some unexpected chromosome rearrangement, which is not desired. So without double-strand break, this can reduce the risk of chromosome rearrangement. It has a very high editing efficiency by single base pair change. And because they don't require double-strand break, so it's accepted for non-dividing cells. While usually the advantages of the technology is also their disadvantages. So deaminase, and the nature of the deaminase makes it having the limitation in their targetable site because they can also target to one base pair. Well, take adenosine deaminase as an example. If the target site A here has many A's surrounding this sequence, there is a chance to generate the bystander mutations. And the size of this base editor is also relatively big. It's above 6 KB. We talk a lot about RNA or DNA editing. So now scientists also want to edit RNA. In 2017, the RNA editors were developed. So it's by the fusion of adenosine or cytidine deaminase with that Cas13. The that Cas13 can bind RNA, but without any mutations there, while the deaminase will be guided to the target site of RNA using guide RNA, and adenosine deaminase can change A to G in the RNA sequence. But this protein is relatively big. So a few years, scientists also developed a more novel technology in terms of they can use the chemically modified synthetic guide RNA to recruit the endogenous ADA, endogenous adenosine deaminase for the RNA editing. And this technology, it only uses a guide RNA to hijack the body's own ADA. It circumvents the limitation from the Crispr-based system that we discussed previously. But the nature of the deaminase give it limitation that they don't have too much of the targetable site. Again, for the RNA, it's temperate. So if a long-term editing is required, then the constitutive expression of this RNA editor is needed. So there are some limitations for the base editing. They can edit the DNA or RNA without double-strand break, but they can only create single base pair change. How can we keep optimizing the technology in order to create more base pair changes? Here comes the prime editing. This is the last technology that I would like to introduce in this nucleus-based gene editing technology session. Prime editing, they have many components in the prime editors. So first, we can see that this structure is like a Caesar, and the target specific sequence is recognized by the prime editing guide RNA. And this will guide the nickase and reverse transcriptase to the target site. The nickase will create single-strand DNA break, and then the primer binding site will hybridize to the sequence, homologous sequence upstream in the single-strand break site. And afterwards, the green color represents the edits that we want. And the reverse transcriptase can incorporate the edits into the 3' flap. These 3' flap include edits desired, but if 3' flap excision happened, this DNA will be converted into wild-type sequence shown in the blue color here, and it will be added again with this prime editing machinery. But if the 5' flap excision happened, it will introduce this heteroduplex intermediate product. They have your desired edit and a wild-type sequence in the same double-strand sequence. The third generation of prime editor, they introduce another guide RNA, creating nick in the unedited strand. So this will trigger DNA repair in order to incorporate the edit in both strands. So far, the prime editing is quite ideal. They can edit this target sequence with multiple different base pairs without double-strand break. That you can also eliminate the introduction of DNA donor within a different construct to carry out gene editing. Because imagine that if you want to introduce a DNA donor sequence with a different construct, And then this sequence need to search the whole DNA genome in order to find the target site. Then the recombination efficiency will be very low. But with prime editing, the guide RNA can guide the nucleus, and the donor to the exact place directly and trigger the repair. So that's why the efficiency of the precise editing is much higher. But the biggest challenge for this prime editor is that, as you can see, they have so many components there. So their size is above 7 KB already. Overall, the nucleus-based gene editing technology have many applications, including improving the traits of plants. And we can also use it to establish reporter cell line and disease cell lines for the drug screening. And we can create the reporter model, animal models or disease animal models to study the efficacy of the drug. And potentially apply that to the patient in order to regulate the gene and correct the gene ex vivo and in vivo. So we talk a lot about nucleus-based gene editing. Now I would like to switch gear and talk about nucleus-free gene editing. Because nucleus-based gene editing, they have a shared risk, which is off-target effect, meaning the guide RNA can drive the nucleus to another location, which is not the desired place. So in order to conquer that, nucleus-free gene editing was developed. And I would like to share with you the technology that we have. It's called GeneRight. The logic behind the design of GeneRight is that we can integrate a therapeutic coding sequence into a specific locus in order to maintain durable transgene expression. For liver-directed therapies, we choose albumin locus as the target site. It's because that, as you can see, albumin is the most highly expressed gene in the liver. And only 1% of albumin is equal or much higher compared to the highly expressed genes in the liver. That means modest rates of integration at the albumin locus is sufficient to achieve near physiological expression level of many disease-related liver proteins. The design of GeneRight is shown here. It includes 5' and 3' homology arm, flanking 2A and transgene sequence. We aim to target the stop codon of albumin so that we can insert 2A and the transgene right upstream of the stop codon of albumin. 2A is a ribosome skipping peptide, which promises the translation of albumin and transgene as two independent proteins. And we would like to target to right upstream the stop codon of albumin. It's because we don't want to disrupt any endogenous albumin gene. This design has no promoter sequence included in order to prevent any desirable activation of any other genes. And the transgene is driven by the endogenous albumin promoter. And there's no nucleus introduced in order to minimize the risk of off-target effect. How does GeneRight work? So the GeneRight cassette is packaged in an AAV capsid. The reason we chose AAV as the delivery vehicle is because so far, AAV is still the most efficient method to deliver DNA into the nucleus. So after the GeneRight DNA with homology arm 2A and transgene is delivered into the DNA nucleus, then it will use the innate homologous recombination machinery to trigger this homologous recombination and insert 2A and transgene right in front of the stop codon of albumin. Then this edited genome is transcribed into albumin 2A transgene fused RNA. With the help of the 2A peptide, the ribosome skipping peptide, the albumin protein and transgene protein was translated into individual protein. And one thing worth noticing is that the peptide here shown 2A peptide is fused with albumin and is secreted into the circulating system. And this albumin 2A is a very powerful, I would say, biomarker to measure and to monitor the GeneRight activity in real time. But as I mentioned that we don't use nuclease, and the GeneRight is depending and utilize the innate cell double-strand break and event, and this efficiency is relatively low. But how can we conquer that? What we have observed is that the liver regeneration drives repopulation of damaged livers with edited healthy cells, its selective advantage. As we can see from the left figure here, the brown color represented the corrected cells with transgene inserted. So 1.5 months, 3 months, 6 months after treatment of GeneRight, we can observe the expansion of corrected cells. And this trend stays the same with albumin 2A also. And we can see that albumin 2A in the circulating system, it's also increasing along with the time. Especially this is in a homozygous mice. One thing worth mentioning that this disease is methylmalonic acidemia, MMA, and we observe selective advantage in this specific disease. We didn't observe selective advantage in a heterozygous mice because the liver is healthy, so they don't have selective advantage. As a result, the expansion of GeneRight corrected hepatocytes leads the disease improvement. In the protein level observed from Western blot, we also observe similar trend that the transgene MUT and albumin 2A increased along with the time in a homozygous after the treatment. And albumin 2A is highly correlated with the liver MUT protein. So albumin 2A can be used as a biomarker to monitor GeneRight activity in vivo. It means that if we treat a patient, we just need to draw the blood at different time points after the treatment and monitor the GeneRight activity. This selective advantage was proved in multiple disease models. In addition to MMA, we also observed selective advantage in A1AT deficiency, Wilson's disease, and the tyrosinemia. This brown color represented the corrected cells, and we can observe the clonal expansion of the corrected cells along with the time. Altogether, it shows that the combination of the GeneRight and the selective advantage can be a very powerful tool to edit the genome in vivo. Now, I would like to give a summary of the gene editing technologies that we covered today. I summarized all the editors that we discussed so far, and the size of different editors determine the package method. Let's take AAV, adeno-associated virus, for example. It has the package capacity of 4.7 kb. If the editors are above 4.7 kb, it cannot be applied for an AAV packaging. And some of the nuclease dependent on the enhancement of double-strand break, but potentially it will also have higher risk of chromosome rearrangement. For GeneRight technology, it's a different case. They don't enhance double-strand break in the cells. It utilize the innate cell double-strand break for this editing event. And the double-strand break determined what type of cells they prefer to be edited. If there's double-strand break, the technology prefer to edit dividing cells. While without double-strand break, base editor, RNA editor, and prime editor, they can be applied to both dividing cell and non-dividing cells. In terms of the complexity of the work, if the DNA sequence is recognized by RNA or DNA, the design is relatively easy. But like zinc finger nuclease and talons, it required amino acid recognition to the DNA, then more protein engineering work is required. Now we talk about the risk. So for the nuclease, dependent on the double-strand break, off-target effect, meaning target to an undesirable site of the genome, is a relatively higher risk. And for base editor, because of the nature of the deaminase, so it might create the bystander mutations. RNA editor, the transient modulation, is its own advantage, because if there is off-target event happen, it will be temporary. It will not last long. But again, if we require the long-term editing, then the constitutive expression is required. For prime editor, I put delivery as a risk here. It's only because, for example, for AAV, the package capacity is 4.7 kb. So it's hard for the AAV to be used for the packaging for prime editor or base editor. But if the lipid nanoparticle is chosen as a delivery vehicle, then based on the current LNP technology, the lipid nanoparticle technology, liver is still the target for LNP compared to the other tissue types. As for GeneRight, the low starting editing efficiency is a risk, but it can be used to treat the disease doesn't require high editing efficiency and the disease with the selected advantage. Of course, all this summary is just at this point, but all these technologies are advancing rapidly. So this summary of the technology pros and cons, it will be updated soon too. At the end, I would like to close my talk with challenges need to be overcome for genome editing delivery. Although we talk a lot about gene editing technology, but it's a long journey from drug development to the treatment of patients. From administration to genome editing outcome, we need to conquer immune response, for example, pre-existing neutralizing antibody. And even if the drug is delivered into the target cell type, there is still a black box in terms of endosomal escape, nuclear entry efficiency, and avoid off-target mutagenesis. The optimization of each of the steps can enhance the gene editing efficiency and the safety for the patient. So this gene editing is such a promising field, and I'm very excited every day being part of it. Thank you very much for your attention. I'm happy to take any questions.
Thank you so much, Jing. We really appreciate your comprehensive presentation. We did receive one question in the Q&A box, and I believe that was submitted prior to the seminar starting. And the question is, what is the status and challenge on using in vitro model to replace in vivo animal testing for IND-enabling studies? Do you have thought about that, Jing?
Yes, we do thought about that. Especially now, there's also the 3R requirement in terms of the reducing the use of animals. I think it's a balance of in vitro and in vivo work, because each of the system has its own advantage and disadvantages. For example, for in vitro work, there was studies showing that it's not translatable from in vitro to in vivo. But it really depends on the in vitro model itself. And then for in vitro, if you do in vitro work, you put your drug directly into the cells without any circulation system in the in vivo. So this is not like exactly mimicking when we deliver the drug to the patient. So at some point, in vitro can give you proof of concept, whether it works or not, but how efficient it is. And in terms of dose escalation study, whether you can translate the dose to the patient is still questionable. But again, there's no promise that the mouse can also be translated to a human, right? So we proved many times that disease can be cured in a mouse, but the translational efficiency in a human is still low. So it's really kind of trying to build up more data in terms of in vitro and in vivo, and hopefully using some AI model to see whether this is predictable or not. And if we can build up more data set, we will have more confidence how reliable this in vitro model can be translatable to human. I think it's just a matter of time.
Yeah, I like your optimism in thinking forward and bringing AI into this. I think that's very interesting. I do agree with you that we will, generally speaking, need more data for each of these platforms to be able to limit the use of animals for IND-enabling studies. I think we're not quite there. And you mentioned dose. I think these studies are important indeed to evaluate editing efficiency, but I would say for in vitro assessments, that's probably pretty good. But I think for safety, in particular for delivery, depending on what you're using, I think it's quite helpful to have in vivo studies for now. Thank you. We're getting more questions, so that's fantastic. I'll go to the next one. How persistent is the correction of the phenotype with GeneRight in liver cells? How do you think about redosing with patients?
That's
so
a
persistence and redosing.
Yeah, that's a really good point. It's a really good question, because GeneRight is utilizing the homologous directed recombination. So once it happened, it will be integrated into the genome. Is in the albumin locus. So far from the data that we observed, we didn't observe the drop of the transgene expression level. It's always with selected advantage for a specific disease model. You can see the enhancement of the transgene expression. So which is a good sign, it's persistent. In terms of redosing, it's a very popular field right now. Well, for GeneRight, we are using AAV. So this is a big question for capsid. So it's not like impossible. There is definitely research published that different capsid can be used as a serotype switch study for redosing. So that's one option. There's other options like you can eliminate the antibodies in the cells before dosing in order to trigger the redosing. So I think many people, including us, are working on that right now. And I still hold a lot of hope for that, for the redosing.
Thank you. And you mentioned that with your gene writing technology, you had low starting efficiency. However, you had a selective advantage. Have you seen in patients, is the selective advantage counterbalancing sort of the limited efficiency that you have? Are you seeing clinical benefit basically, or is it too early to tell?
I think for us, we do have the GeneRight put into the clinical trial already. And then we monitor the patient using albumin 2A as the biomarker. And we are very happy to see that we observe integration event. We observed albumin 2A expression in a patient. It's the published information. And we also observed the increase of the albumin 2A along with the time. But for the patient, it's different compared to the mouse. The liver regeneration efficiency in terms of the lifespan is different between mouse and patient. So it takes much longer for these build up transgene expression in the patient.
Thank you. Another question from the participants. In your scientific opinion, which editing technology seems to be the most promising in regards to allogeneic cell therapies in solid tumors? It's a very specific question.
Solid tumor, what's the other one?
So I think the question was about using allogeneic gene therapy for solid tumors, and which technology, genome editing technology, do you think would be most promising in that setting?
Wow, this is a very tough question.
I was going to say, if people had the answer, maybe we would know already, but it'll be interesting to know what you think.
So for the tumor, it's a very complicated system, to be honest. And if you want to correct specific genes, it really depends on the mutations of the cause too. So solid tumor, you're not able to use ex vivo method for the delivery, and then in vivo delivery is the method. Then it comes with the delivery. So whether we can use a local injection or is a systemic delivery. It also depends on what tissue type too, right? So it's a mixture of delivery and the technology. To be honest, I don't have a yes or no answer which one is the best, because it really depends on the disease itself, like which tissue type and what type of delivery system that you can use in order to choose what method can be applied. Each technology has its own unique niche for the application for the specific disease.
Yeah, I think it's fair to say that if you need to integrate the whole car cassette, it sounds like the technologies that you had later in your time chart are probably more appropriate, right, to integrate a large payload. And I agree with you, delivery issues are certainly going to be key here. A couple other questions. I think we still have time for this. Do you also see immune activation using the DNA sensing pathways that later affect the fate of the transduced liver cells?
Can you repeat that question again?
Sure. Do you also see immune activation, and in parentheses it says DNA sensing pathways, that later affect the fate of the transduced liver cells?
I see, whether there's any immune response to the corrected liver cells. It really depends on the transgene itself. So for the mice, it's a common trend that we observed that if we introduce another species of the transgene into the mice, and it might trigger immune response. At the beginning, the cells can be corrected, but the immune system will attack the cell because they don't recognize that protein and see it as a foreign protein and attack the cell. So you can see a drop of efficacy. But if you use the same species of the protein, and we didn't observe any issues for that. It depends on whether these two species sequence are homologous, are very similar to each other.
Great. So it sounds like that hasn't been an issue in your experience. Thank you. One more question. Would you know if there is a good correlation between in vitro and in vivo off-target evaluation for system with Nucleus? So correlation between in vitro and in vivo off-target evaluations.
Yeah, it's a really good question in terms of what system we should use to evaluate off-target. But as we have known so far, in vitro is still not that predictable. It doesn't align that much compared to the in vivo off-target evaluation. So I still believe that it's better to do both in vitro and in vivo, so we can accumulate more data set and see which one is more predictable, to be honest, because we also don't know whether in vivo the mouse is predictable for human too.
Yeah, I think biodistribution here is important to be evaluated in vivo, right? So it's the type of off-target effect that you're going to have, but also which tissues, where is that happening? So I agree with you.
Yeah, exactly. And then the dosage of in vitro is also critical, because as you know, if you put more nucleus in the in vitro, you definitely will increase more off target, right? So it's very tricky what MOI you should use for in vitro.
Yeah, that's a fair point. I've heard of colleagues who use that for signal detection to see where to look for other technologies. Great. So I think we covered all the questions. So maybe you want to pause for a couple minutes to see if there are any additional questions from the participants. And otherwise, Jane, I definitely want to give you a big thank you for volunteering to present on behalf of BIO for this seminar. I really enjoyed your presentation and the overview of all the novel technologies that have existed and are emerging in genome editing. And we really appreciate everybody's participation and everybody's questions. So maybe, Jane, I'll turn it over to you if you want to say a few words.
Sure. Well, thank you very much for organizing. It's a great opportunity to share and also to go through the very high level in terms of the technologies here, but apologize that cannot cover all, because it's such a blossom in terms of the development of the technology. So only cover some key players here. I think Nucleus still holds a great future if we can eliminate the off-target, understand the off-target, and eliminate the off-target effect. At the same time, while we are optimizing the nucleus, we can also utilize some nucleus-free technology as GeneRight, because it can be treated as any traditional gene therapy vectors, like AAV-based gene therapy vectors, because it's promoter and nucleus-free.
Thank you so much, Jane. And everyone, have a great rest of your day and a great weekend. Thank you.
Thank you. Bye.